Donna, I have a question that was submitted specifically for you. This person, Mrinal Shah, I believe is this person's name, from AbbVie. They're asking, you mentioned that in the manufacturing process, you are able to react based on the outcome of the prior step, but would seem like a process change when compared holistically to the overall process. How would regulatory bodies react to a process that changes ever so slightly from batch to batch?
I've found no issues with it so long as you present the data. And it goes back to what I just discussed, understanding what contributes. We talked about quality by design. So you've got your CQAs, you've got your CPPs, understanding what contributes to that success that you desire, and knowing that there's various things going to cause variability in that. I mentioned having those in-process testing, cell counts, population doubling, measurement of metabolites to understand different cells. Every patient's T cells doesn't come in the exact phenotypic profile. Some may have more memory cells, some may have more, they just vary. All T cells are not created equal. Let's just put it that way. They are going to come in, the patients can come in with different growth curves. Understanding what contributes to that, you can have the same patients and you can have a process. You do this on this day, you do this on this day, you supplement feed on this day, you supplement feed. So you have your base process. But if you see that you're using lactase much faster than necessary, those cells are going to die on you, or you're going to start having apoptosis occur, and you're not going to get the yield that you expect. So the agency is quite amicable to you developing those algorithms. If I see this, then I do this instead. It's just a matter of doing those studies and understanding your process. Again, a lot of that can even be established with various healthy donors. All healthy donor T cells do not grow at the same rate necessarily. There's a pattern that fits 90% of the time, but you're going to have outliers, and understanding what to do with those outliers. If you need X number of cells to meet a patient dose, that you maintain that and you don't compromise that growth of those cells along your culture process. I've found it quite amicable to work with the agencies. They're more amicable to building that into the process. It's just having the data to support that.
Sure.
Hope that helps.
Certainly does. All right, I think we have time for one more question. I'm going to direct this one to you. Could you please elaborate more on internal process control and how does it work?
Internal process control. Oh, sorry, go ahead, Donna.
Go ahead. No, go ahead. So sorry.
Oh, I was just saying, maybe it's me, but I want to clarify that question. I think this might be referring to the unit operations process control that we talked about at the very beginning of the hour, where what I was suggesting was to break down the end-to-end patient or the product journey and identify those unit operations, those pieces of the process that actually you can try to control better and aim to reduce variability, right? There are multiple ways you can do that. I know we're running out of time as well, but I will tell you there are. Let's take an example of you get the raw material cells from your apheresis from the patient and you begin manufacturing, right? If you are able to control how you're going to be thawing out that cryopreserved cells and activating those cells, and you have a very controlled process for it. And I'm aware of a company that Carl June started called Blue Whale, and that's what they're trying to do. They're trying to actually control exactly how that specific step is controlled. Like not before, not after, but they just focus on that one little step. Similarly, there's another company based out of the Bay Area. I'm forgetting the name. I think it's called Cellular Vehicles, something like that. They are only focused on the final drug dose formulation and administration, right? So back to what I was saying, if you were to like piecemeal the entire product journey and identify what parts of those processes can be controlled, can be managed, that is where you can try to reduce variability from that individual step and reduce the contribution of that variability for your end-to-end journey. And if this doesn't answer your question, happy to take it offline as well if need be. But Donna, you were saying something before I interrupted you rudely?
I think it's a concept that's probably not stressed as much. You buy kits and they have controls, and in a clinical lab, there's controls for X, Y, and Z. There's not established, of course, we're working with live cells, and having controlled live cells is a bit difficult. That's why I default to, you can learn a lot by establishing early on a bank, a cryopreserved bank of PBMCs to use in assays, cryopreserved bank of your target material. And we enrich for CD4, CD8s before we go into culture. So we have a preserved bank of healthy donors. We know what those donors do. So when I introduce a change, I can go back to those donors. I don't have to do side by side. I have an established expectations with those healthy donors. I do the healthy donors with a new process or change in manufacturing devices, and I show the comparability or improvement. That's the same thing that applies with assays. I think that is a bit more difficult, especially in the functional assays for cell therapy, when you're trying to understand or really define your mechanism of action, develop functional assays that'll correlate ultimately with clinical outcome and provide you with an efficient potency assay. It's like, how do I know that assay is performing consistently across the time and across different iterations? So again, if you have PBMC banks that you can put in, effectively thaw and controlled thawing, and our product that you can put in those functional assays to say that they do the same thing every time. So when you do patients versus your healthy donors, you know that your assay is working. So that difference from what you see in healthy donor results in manufacturing RNA assays versus the patients, you can better define what's going on and how to adjust accordingly. I found that extremely useful. It's a little bit of an effort, takes up liquid nitrogen space, but those internal process controls and mining that data set is hugely important and can help. Again, it is about breaking it down to those unit operations. What I expect at this stage, what I expect at that stage. And it goes a little bit with more that upstream, downstream processing mindset that blends well, quite frankly, with investors. They can relate to that better than they can this, okay, we put it in a pot, we come out with this type thing. So breaking it down to those unit operations, having the necessary controls to know that as I do this, I get my expected results on something that I have well-established results for. So that's how I use the internal process controls.
Yeah, and Donna, I would add that this internal controls applies to not just the process, but the people and the technology that you use as well.
Yes.
Right. So it has to be a holistic thing. Exactly. Right. And we've had examples where we have certain internal control processes that can be performed by RPA, like robotic process automation, versus doing it manually. And we've actually just stepping away from CGT and like the manufacturing process itself. These are common use cases where they use data-enabled risk and control analytics in other areas like finance, procure to pay, order to cash. So it's most mature there, but I think we can take some learnings from that side of the industry and apply those practices here. Just my 2 cents on this.